I'm going to be discussing microfilament polymerization and also depolymerization and the different proteins that are involved with these mechanisms and basically how they work together, but I'm going to be taking it in a little bit of a different direction. There are a variety of proteins involved with the different mechanisms, and they basically can be grouped according to their function. And the way I like to think of it as actin is a big high school, and these proteins are basically different groups of proteins that kind of go into their own individual cliques according to function, and that's kind of the theme that I'm going to use throughout this video. So this first clique that I'm going to be discussing are basically the proteins that are involved in regulation of polymerization, and these proteins are basically going to be in charge of regulating the polymerization and the growth of these actin subunits. This first protein that I'm going to talk about is thymosin, and basically what this one's going to do is it's going to bind to different subunits, and this binding in turn is going to prevent the assembly of the actin filament, and altogether it's going to inhibit polymerization altogether. So what we're going to do is we're going to give this protein some mean eyebrows so that it can kind of fit its character. The second protein that we're going to be talking about is profilin, and this one is sort of the same thing. It's also going to bind to subunits, and it sort of acts as a catalyst in that it's going to speed up elongation as well. Well, it's actually just going to do the opposite, and in this turn it's going to promote polymerization altogether. Up next we have the nucleating sort of protein. So right here I'm going to draw an actin subunit, the minus and the plus end. So over here on the minus end, I'm going to start off by drawing the ARP23 complex, and this complex is basically going to bind to a minus end, and it's going to form a web and basically help nucleate the assembly of more actin filaments and sort of promote branching as well. On the plus end, now we have formin, which is also going to bind over, and this one is basically going to just remain associated with the plus end and do sort of the same thing, just from the opposite end of the microtubule. So these guys literally work together in order to show that branching that you see in a lot of microscopy images and such. Next, we move on to our second clique of proteins, and these proteins are going to be in charge of controlling filament dynamics. So sort of thinking of treadmilling and catastrophe and sort of that diagram that's commonly shown in most textbooks. The first one that we're going to start off with is called tropomyosin, and basically I'm drawing another actin filament right here, and tropomyosin, given it some strong arms as you can see, is going to stabilize that actin filament. It's really important because this stability is important for a lot of different cellular processes. So yes, our next protein that we're going to talk about are called capping proteins, also known as CAPZ. These snazzy little proteins are going to bind to the plus end, and they're basically going to prevent assembly this way, which is really cool. So basically this cap protein can couple with other proteins in order to make a lot of important processes happen. So yeah, our next protein that we're going to talk about is called tropomodulin, and this one is going to bind to the minus end of the actin filament, and this is basically going to prevent disassembly as well as assembly. And basically the effect of this protein, it's going to reduce the pool of actin that is within the cell, thus reducing the rate of actin growth, which is really important because it also helps in a way to regulate and control the actin filament itself. Third clique that we're going to discuss is in control of filament depolymerization, also known as the filament shrinkage. And the first one is really, really cool. It's called gelsolin, and this one is super interesting. So we're going to start off by drawing the actin filament, and the reason why I drew it this way is because this protein is going to sever the filament in half, and it's going to bind to the plus end. And the effect of this is a little bit groggy, and that's because it's going to be coupled with the capping proteins, which is going to in turn not help polymerization at all. And due to the capping protein being on the plus end, even though we have a new plus end, it's not really going to do anything. But this also is why it helps depolymerization and is in this group of depolymerization, because it makes a new minus end as well, and this is going to help facilitate and encourage depolymerization of the actin filament. So yeah, and that's pretty much it for this protein. Super cool. I'm going to go ahead and draw in the capping protein to kind of show, there we go right there, that's why we don't have polymerization. There's also another protein in this group called cofilin, which we talked about earlier. Cofilin's really friendly. It likes to be in more than one click. And basically this one's also going to help facilitate depolymerization in its own way, because it basically accelerates the disassembly and depolarization. So click number 4, this one's going to be in charge of filament structure. The very first one that we're going to talk about is filament, and filament is a type of splicing protein. And what this is going to do is going to go ahead and bind two different little branched actin filaments, and this is going to go ahead and stabilize the microfilament altogether, which is just really awesome for different cellular processes such as like cell motility. In addition to this protein, we also have alpha actinin, and alpha actinin is heavily involved in cell motility in that it is going to help with focal adhesions and contacts, which is really, really important in cell motility itself, in attachment and such, for when a cell is doing movements and such. So these two proteins together are just going to help move together and kind of help that process all together with cell movement, and structure is really important in that. And that's all I have for my actin filament polymerization. I hope that you learned a lot, and thanks for watching.